Let's go ahead and get started. My name is James Peterson, and I'm the Senior Director of Operations and Outreach for the Center for Physical Genomics and Engineering. And I'm here to introduce the speakers today for the Physical Genomics Predoctoral Training Program, or PGTP Research Showcase. The PGTP is a NIGMS-funded program with the mission to train the next generation of transdisciplinary scientists that will bridge molecular biology, bioengineering, physics, optics, chemistry, and medicine. We want our trainees to be able to speak fluently across multiple disciplines. For instance, if you're in a lab that focuses on advanced optical imaging, we also want you to be able to speak about molecular biology techniques and gene expression, and possibly how that affects the understanding of treatment of disease. For those of you that may be new to physical genomics, it's a new field that aims to understand from the first principles of physics the interrelation between the structure and function of the genome, or more specifically, the role the three-dimensional structure of the genome has in generating global patterns of gene expression. Dr. Backman, the director of the center and training program, likes to use the analogy that if the genome or DNA is the hardware and epigenetics is the software, then the three-dimensional structure of the genome or chromatin would be the operating system, which then enables global patterns of gene expression and ultimately cell phenotype. So, as I said, Dr. Backman is director of Center for Physical Genomics and Engineering and the PGTP. He is currently on vacation, so I don't think he was able to make it today. But I, along with Bob Benking, the senior program administrator of the center, help administer the PGTP. So we have 42 faculty from 3 schools and 17 departments across Northwestern. We currently have 16 trainees. Actually, Raymond Fang is graduated, but he's still part of the program. So he's in his, actually entering his 4th year in medical school. So that's great. And we are currently deciding on some new applicants, and hopefully we'll have that done shortly. So, without further ado, go ahead and introduce the research speakers for today. Paola Carrillo Gonzalez is a student who's co-mentored, who has dual PhD advisors, Szleifer and Vadim Backman, and her transdisciplinary co-mentor is Mazar Adli. She'll be speaking on the impact of charge regulations and ionic intranuclear environment on the nucleosome core particle. John Carrillo is co-mentored by Ji and Vadim Backman. Those are his PhD advisors, and his transdisciplinary co-mentor is Gemma Carville. He'll be speaking on deep learning modeling of RNA translation initiation in human cells and diseases. And finally, Tiffany Kuo, who is advised by Vadim Backman, and her transdisciplinary co-mentor is Yogesh Goyal. She'll be giving a talk on exploring the role of heterochromatin protein 1 alpha in stabilizing chromatin packing domains. So we'll go ahead. I'll also say that monthly seminars of physical genomics upcoming speakers. So every month we have a speaker on physical genomics here at the Center for Physical Genomics and Engineering. This is the upcoming speakers, and these, if you want to connect with us, please find us on YouTube, X, Blue Sky. But without further ado, I'll go ahead and stop sharing and let Paola get in on the action.
Ooh, it says I can't share yet.
Yeah, I know, because it, I need to, maybe, where'd you go? Oh, there it is. Okay, sorry about that. Go ahead, Paola, take it away.
Cool, let's see. Okay. Am I sharing the right screen?
Yes, that's
Okay.
the presenter screen. Okay.
Perfect. Okay, so yeah, let me get started. Good afternoon, everyone. My name is Paola Carrillo Gonzalez. I am part of the Backman and Szleifer labs, and I'll be talking to you today about my project and on chromatin, and more specifically, how the intranuclear environment affects chromatin's charge and structure. So I know James recently, right now, talked about physical genomics, and we might all have a background, but I just want to lay down the background that's necessary for my presentation. And so, of course, I want to start off by talking and describing what chromatin is. And chromatin is a complex of DNA and histone proteins that somehow condenses to form chromosomes in our eukaryotic cells. And our chromatin and chromatin organization ultimately dictates what genes are expressed in our body. And specifically in cancer, for example, irregular chromatin organization is often found, and it's a characteristic of cancer. So it's something that definitely we are interested in to try to see what is happening there. We can also view chromatin as a polyelectrolyte, and this is because we have charges coming from the DNA and the histone. The DNA has a negative phosphate backbone that interacts with the positive histone. This histone specifically has four intrinsically long disordered tails, and it's these tails that mediate interactions between nucleosomes and other proteins to get these higher order structures like a chromosome that fit eventually in the nucleus. But with having such a charged system, it offers a question of how is the structure mediated while encountering all these electrostatic repulsions coming not just from the histone and the DNA, but also all the ions that are in the environment. So something that's really interesting. And we see that ions are having a profound effect on the organization at different length scales. Specifically in the DNA and nucleosome length scales, we know that there's a charge distribution happening. And so the charge regulation of the negatively charged DNA chain shows a strong preference for binding of cations like sodium to the DNA. And when we go into a higher order structure, we do see signs of compaction influenced by monovalent and divalent salts. Because of its divalency, divalent salts have been seen to have more of an effect on chromatin compaction. But also, several experiments have actually demonstrated that the ionic environment plays a role in selecting phenotype. And in this study specifically, they injected different concentrations of magnesium chloride into mouse embryonic stem cells, and they observed the large change in differentiation. So this is just showing how ions are really dynamic and influence all parts of the system. And that really brings me to my big question that I want to answer throughout my PhD. And this is, what are the guiding principles and the mechanistic function of ions in chromatin organization? I tackle this question through both experiments and theory, but for the purposes of this presentation, I'll only be focusing on the theory side. And so studying chromatin, whether experimental or computational, is hard by nature, given how tiny and dynamic the system is. In traditional modeling, we have all atomistic modeling. Um, which gives us a very detailed representation of a system. But of course, this limits how large our system can be. And not only that, but with this all atomistic modeling, there's also a time scale limit. And so in simulations, the system may not reach equilibrium, and that can also lead to incorrect conclusions. So while we are able to get very good data from all atomistic modeling, for me, where I want to model a larger system and all their interactions, it's not going to be a as useful. We can also do the other traditional modeling, which is a coarse grained model. And so this, of course, allows us to simulate larger systems, but it requires setting certain parameters that I'm particularly interested in, such as the electrostatics. More specifically, you need to incorporate fixed charges or fixed pH, and these are all part of charge regulation that I'm interested in. So unfortunately, coarse grain modeling won't work for me either. And experiments are also good because we can also measure the actual chromatin behavior, but we can't necessarily see the structure or know more about the thermodynamic properties of the structure. And so to actually study how the ions are affecting chromatin, I use an in-house developed model known as molecular theory. And molecular theory, as I mentioned, is in-house. And it's a statistical mechanical model that is able to produce thermodynamic and structural information, such as free energy, charge distribution, local concentrations, and more. And it does so by solving a really complex free energy functional, which takes into account the properties of a polymer system. You can see all of our properties in this box here with the free energy functional. And you can see that we take into account the conformational entropy, the translational entropy of all the mobile species, the chemical equilibrium, electrostatics, interactions. And so it's a really complex function that really is the ground and the core of molecular theory. We can modulate the environments and the charge, such as the charge and size of the molecular species. And then the model solves this complex free energy equation, producing local system information, like the local proton concentration. And so in molecular theory, the DNA is represented by a triplet of atoms, specifically a sugar, a phosphate, and a nucleotide. And the histone core, on the other hand, is represented by atomistic representations of the amino acids. So we actually have all the little atoms involved in each amino acid. And there are a number of reactions happening with this as well. We have phosphate chemical reactions. We have the acid base as well as all the monovalent and divalent salts interacting with the phosphates. And we also have acid-base equilibrium with the amino acids, as I've mentioned, and ion condensation reactions as well. So we have a large number of reactions happening. And what we were able to find when we included all these reactions is that the nucleosome is very heterogeneous. When we look at a single nucleosome, we see specifically the electrostatic potential difference between the DNA and the histone. Now, you can see there's a relatively purple halo around the screen structure, and this screen structure actually is the amino acids. So a very large difference between the actual core and the DNA. But what is actually really interesting is that there's also a difference in local proton concentration, and this is very the difference is actually quite large. We see this amino acid core reach a pH of around 9, while the DNA is encountering more of a pH of around 6.5, all of which are very different from the bulk solution. So you can see like the lattice is a different shade of blue in comparison to the DNA and a very different color than the core. So we can set environmental parameters, but we can see that there's very much a local heterogeneous distribution of the electrostatic potential and local proton concentration among many more. And the beauty of molecular theory is that we can again change these concentrations and see the effect that magnesium, for example, has on the structure. And magnesium is particularly important because it's one of the most prominent ions that are known to affect the chromatin organization, among, of course, other processes. But when we increase magnesium in our results, we see that this purple line that is depicting the negative charge of the phosphates is actually getting lighter and lighter. And this is showing that as we are increasing magnesium concentrations, we're neutralizing this negative DNA backbone. And this is, of course, going to have consequences on the higher order structure. Um, with molecular theory, we can actually quantify all of these parameters. So we have pretty images and we can actually see the numbers. And so we can look at the fractional binding as well as ion excess. And basically, this is the different binding states that phosphate has with all of the protons, cations, and we can also quantify the counter ion cloud that is happening outside the structure. And one of the interesting things that we found when we include physiological conditions is that as in millimolar ranges, we do see that approximately 55% of phosphates is bridged with a divalent magnesium ion. And this is extremely interesting because it's saying that almost if we were to extrapolate this into a higher order system with a higher density, this would mean that a lot of the phosphate is actually bound with magnesium in the physiological concentrations and conditions. Um, and so as magnesium is as magnesium is binding, it's dominating the overall reactions, which is why you see this red line really take off between these lines. And this is coupled with, of course, the counter ion. So as magnesium is increasing, we also see that there's an attraction of potassium and a depletion of chloride in the counter ion cloud. And again, these are all going to have significant effects on the actual reactions that are happening within the nucleus. Um, and so it's giving an indication of how this higher order structure is going to be mediated by these local parameters. I hope I didn't go too fast. At the moment, we are expanding the system. I just showed data for a single nucleosome core particle, but we are working to expand to a higher order system with a set of tethered nucleosome chains, and this would mimic more of the higher, denser um, density profiles, and specifically those relating more to lamina attachment or domain interfaces, because it is tethered to a surface. So we're working on getting that running, and it's the results that we see with the one nucleosome are only going to be further exacerbated with this chain of nucleosomes. That is going to be really interesting to see. Not only that, but we also know that the nucleus also involves a lot of different proteins. And so we eventually want to get these proteins in the simulation as well, so that we can see the effects that the proteins and maybe the enzymatic rates have. So all really good things coming. Um, I want to thank, of course, my advisors for all the help that I've had throughout this project, and of course, all my lab mates. And yes, of the T32 physical genomics training grant as well. Um, yeah, I am open to any questions. I hope I didn't go too fast.
No, you did great. If you guys want to ask any questions, you can put in the chat or go ahead and raise your hand. I believe you all have the ability to unmute. Well, in the sake of time, since we got started a little late, if we can come back to questions at the end as well. So just to kind of keep things moving along, we'll go ahead and uh, have Tiffany give her presentation next. But thank you very much, Paola.
Perfect.
Okay, you guys can see my screen.
Yes.
Okay. Hi everyone. My name is Tiffany. I'm a rising fifth-year PhD student in Dr. Vadim Bachman's lab, and today I'll be talking about my project that focuses on exploring the role of heterochromatin protein 1 alpha in stabilizing chromatin packing domains. So I wanted to first give a brief overview of kind of the scale at which we're looking at. So the human genome is organized at multiple levels. So we're starting out from the DNA helix, and this is wrapped around our core histones to make up nucleosomes, and that's further folded into the chromatin fiber, loops, chromatin domains, as well as TADs. And this is all folded up nicely into chromosomes to fit into the nucleus. And so there's kind of an interplay of principles that leads to kind of these major emergence of major organizational features of the genome. So kind of like this graph that really showcases the various dynamics that are happening within the nucleus. So we have polymer-polymer interactions, chromatin dynamics, phase separation, as well as architectural elements where we're looking at the nuclear lamina specifically, as well as the oscillation of genes going on and off. And so in Vadim Bachman's lab, we primarily are studying the supernucleosomal scale, where we're looking at how this, essentially how the human genome being packed into this hierarchical manner, how does it facilitate gene expression and overall genome function. So in previous work that has been done in our lab, we use chromatin scanning transmission electron microscopy, or chromstem for short. And we've been able to identify these chromatin packing domains at a very high resolution. However, this technique is very tedious as well as time-consuming and is very laborious. So in our lab, we primarily use two main techniques or imaging modalities to probe chromatin structure. So on the left here, we have partial wave spectroscopic microscopy, or PWS for short. This is a great technique because it's very high throughput. It's label free, which is great, so we're not messing with the cells. And we're able to do this in both live or fixed cells. And this allows us to probe structures between 20 to 200 nanometers. On the right, we have a very big technique that we use in the lab, stochastic optical reconstruction microscopy, or STORM for short. This is a fixed cell technique, so that is a downside that we're not able to look at epigenetic modifications in live cell. However, we are able to localize these epigenetic modifications of interest at less than 20 nanometers of resolution. And so some of the information that we can extract from each of these techniques really help us to formulate our understanding of chromatin packing domains and their function. So with PWS microscopy, with both in fixed and live cells, we're able to extract chromatin packing scaling factor D. This is essentially a quantification of this relationship between the chromatin chain and how it's fitting into three-dimensional space. With live cells, additionally, we're able to kind of capture the dynamics of chromatin structure. So we're able to extract fractional moving mass values, which really provides us insights into the amount of chromatin that's moving. Furthermore, we're able to look at effective diffusion, which really just gives us more insights into the mobility of the chromatin. With STORM, we recently have developed this cluster analysis algorithm, which allows us to examine the spatial organization of these marks of interest. So in this figure on the bottom right, we have a representative kind of like image of this cluster analysis where we're able to look at euchromatin marks and active RNA pol2 and how they're localizing around this heterochromatin cluster. And so furthermore, in our lab, we've been able to look at how the functional aspect of these packing domains. And so in a recent work published, we were able to look at co-localization of packing domains with PWS and where RNA polymerase II is localized with STORM. And we've been able to see that active transcription is very much occurring at the periphery of these packing domains. And with recent work in the lab, we've been able to further kind of develop our understanding of packing domains, how they're formed and how they're maintained. So on the left here, we have a modeling of the sizes of these euchromatin and heterochromatin enzymes. We saw that these euchromatin enzymes are much larger in size compared to heterochromatin enzymes, therefore allowing for preferential penetration of these heterochromatin enzymes into kind of these more dense cores of these domains. And then on the right here, we have kind of an outline of how we're looking at how these domains are being formed and the functional outcomes. So kind of the first rule that we're looking at is kind of the creation of these nascent or new domains. The second one is kind of an emphasis of the size-based diffusion that stabilizes the core of these domains. And then finally, we're looking at how the transcription is happening in these optimal conditions. So that kind of brings me to the focus of my work in this presentation today. I'm really interested in looking at the function of H1 alpha as a crosslinker to maintain the structure and organization of chromatin packing domains. And so we acquired this oxidant-inducible Degron system that specifically degrades H1 alpha from Dr. Andrew Stevens. It's a great system because it really allows us to rapidly deplete the protein, and it's also a reversible system. And so as we know that H1 alpha is a crucial crosslinker, it's been very much studied. We wanted to see how the degradation of this protein would affect stabilization and maintenance of packing domains. So we started out our research with using PWS to explore H1 alpha's role in maintaining packing domains. So on the top here, we kind of have a spatial map that really showcases the chromatin packing domain variations across the nucleus. So you can see visually there is a bit of a difference just looking at the D maps. when we're looking at the specific values that we can extract to kind of better understand generally, on average, what is happening to chromatin in these cells, we see that overall chromatin packing scaling D slightly decreased. There isn't too big of a drastic difference. However, we do see that there is an increase in fractional moving mass and a decrease in effective diffusion, hinting to us that the loss of this H1 alpha crosslinker is potentially leading to the expansion or swelling of these domains. However, the limitation of this technique is really that we're looking at an average nuclear D as well as average measurements across the population. So we wanted to use STORM to better understand the distribution of packing domains upon degradation of H1 alpha. So the two main marks that we use here to understand, to better uncover the composition of these packing domains is first EDU, which is a nucleoside analog of thymidine and is incorporated into DNA during active DNA synthesis. So we're really able to capture higher density DNA regions. And then we also mark for H3K9 trimethylation, as I mentioned before. So H1 alpha is a crosslinker for this K9 mark. And so this is kind of our proxy for measuring these dense domain cores. So when we look at this visually, there is a pretty big stark difference between the K9 marks in the 5-hour oxygen compared to control. EDU, there's also a slight decrease in the number of localizations. Just looking at this visually is not going to give us too much information. So we use the cluster analysis algorithm that we have in our lab, and we quantify the radius of these domains. So essentially the size of these domains by looking at or measuring the distance from the center of the cluster to the boundary. Also looking at the cluster density, which is measuring the number of localizations per area. And looking at kind of the trends here, we see that with the K9 mark, we have this increased size, but a decreased density without H1 alpha. So it's becoming larger and more diffuse. With EDU, we see a slight decrease in domain radius and an increased density without H1 alpha. So here we're kind of looking at generally what is happening. It seems that H1 alpha is very much this core crosslinker that's potentially acting like glue to maintain these packing domains that we're interested in investigating more about. And so kind of the future work and work that we're currently working on is really adding kind of another layer of exploring the role of ions via magnesium chelation. And so with Dr. Szleifer's group, we are interested in potentially seeing if we can model the varying crosslinks for both H1 alpha and ions at these various or different timescales that they're acting upon and how they affect packing domains. And another aspect of my work that's kind of missing right now is really understanding the transcriptional output. So we are labeling RNA synthesis for H1 alpha degraded cells and pairing this with the RNA sequencing data to better understand what is happening to the transcriptional changes when we're degrading H1 alpha. So with that, I wanted to thank my lab for their support, as well as collaborators, as well as the NIH T32 Physical Genomics Training Program for the funding these past 2 years. Happy to take any questions at the end.
Thanks, Tiffany. Anyone with questions? Yes. Feel free to put them in the chat, or it seems that you're able to unmute if you would like. So Tiffany, what's your plan to work with Yogesh? Will he be able to help you at all with some of the RNA work?
I think potentially with another project we could
Okay.
work together. I think for this one I have most everything, but I know that Yogesh works a lot with fish, so I think potentially we could collaborate on that as well.
That'd be good. I'm sure he has a lot to offer. All right, we'll go ahead and keep moving forward. John, if you'd like to go ahead and share your screen, we can. We're moving right along. This is great. You guys are very efficient.
All right. How is this? Can you see
That's the
my
good one.
screen in a?
Yep.
Okay. How do I hide the bar again? I don't know the shortcut for that. Hi,
We don't see it actually.
I got it.
I don't
From
see
my
the
screen, I
bar.
was being selfish. All right.
Yeah.
All right. Thanks. So I'll be presenting a project called Deep Learning Modeling of RNA Translation Initiation in Human Cells and Diseases. So my project deals with translation, although the first two projects you heard were about transcription and specifically epigenetics. I look at the genetics of translation, so a little bit opposite. Here I have a slide talking about, a very biology textbook definition of translation. So I think many of you are familiar with this, but I just want to make sure we're all on the same page for terminology throughout this talk. So here I have kind of a crude drawing of an mRNA transcript. We see the 5' UTR, the coding sequence, and the 3' UTR. In translation initiation, the 43S pre-initiation complex scans 5' to 3' down the UTR with one of the ribosomal subunits and initiator tRNA until the AUG start codon is reached. At this point, the ADS ribosome is formed and scans one codon at a time down the coding sequence, producing the protein product associated with this transcript. This coding sequence is called the main open reading frame. Any sort of translational product is called an open reading frame, or ORF for short. And this AUG start codon is called the canonical initiation site or annotated initiation site. Now, there's a sequencing technique called global translation initiation sequencing that allows us to identify locations of translation initiation throughout the genome. We use two molecules, cyclohexamide or lactimidomycin, to arrest ADS ribosomes. Cyclohexamide is a small molecule. They can arrest ribosomes at any point of elongation, whereas lactimidomycin is much larger, so it only arrests ribosomes with empty E sites. These would be initiating ribosomes. By identifying sites of lactimidomycin enrichment over a cyclohexamide background, we can identify locations of translation initiation. Now, as studies on RNA translation have become more popular and have grown in complexity, it has become clear that transcripts not only possess this one single location of RNA translation initiation, like I showed you on the first slide. In fact, many transcripts possess multiple locations of translation. We'll call these additional locations non-canonical translation. They can occur upstream, downstream, overlap with the canonical site, or be on non-coding RNA strands. And although this is extremely prevalent, we do not know much about why non-canonical translation occurs at the positions it does. So we want to learn more about that. We can use this sequencing technique that I explained to identify locations of this non-canonical translation. So here I have a potential mRNA transcript. Here's the annotated initiation site, and we see this lactimidomycin enrichment over cyclohexamide background. Okay, that is great. But now if we look up in the untranslated region of this transcript, we see there's another AUG codon that shows lactimidomycin enrichment over a background, indicating an upstream open reading frame. Furthermore, this GTI-seq published study also shows that not only does AUG initiate translation, but so does CUG, GUG, and any single nucleotide variant of AUG. So these other codons can initiate translation, and there's other literature out there that supports that CUG, GUG, et cetera, can initiate these translational events. So I hope that so far what I've shown you all is that translation is a much more diverse and complex process than I think many people originally thought or think of it as. And some of the features that would affect translation initiation are encoded in our genetic sequence. And they would be the type of start codon, the length of the 5' UTR, the presence of upstream open reading frames, and the genetic sequence context. So what are the nucleotides directly around a start codon? In eukaryotes, it's commonly known that the Kozak sequence is the genetic sequence that produces optimal translation initiation. So while we know that these features do exist, we don't really know how the features quantitatively impact translation. What are the strongest features? And we definitely don't know how these features interact with one another. So computational models are needed to systematically quantify how the genetic sequence influences translation initiation. I'm going to argue that using deep learning models is a smart idea to understand how these different features exist and interact with one another. So we can take a potential genetic sequence, pass it through a deep learning model, and predict the rate or strength of translation initiation at a potential start codon. Through the operations of back propagation and gradient descent, we can sort of optimize this model or the weights in this model to identify and relate the strongest interactions between different features present in the genetic sequence. And this added flexibility is what we need to learn these new novel features. People have tried this method before. However, there's been some shortcomings. So initial deep learning classifiers use short genetic sequences and are unable to contextualize start codons within their environments on full-length human transcripts. More recently, deep learning models are trained on synthetic 5' UTRs, so their input is much more complicated and a longer genetic sequence, but synthetic data has features that are not really prevalent in human data sets. So they're missing on the important features that drive translation in humans. So therefore, my solution is that I develop multiple deep learning models to observe translation initiation at different resolutions in human transcripts to account for non-AUG start codons, full transcript genetic sequences, and features present in human sequencing data. We could break this kind of work down into 3 aims. So first, we develop a deep learning model to look at local features nearby start codons. Next, we can train an expanded deep learning model to visualize translation initiation probabilities across entire human transcripts to get that added context. And then finally, we can identify clinically relevant variants throughout the transcriptome that affects translation initiation. Starting with our first deep learning model, we can learn what genetic features exist really close to start codons that would drive individual AUG or single nucleotide variants of AUGs to initiate translation in a canonical and non-canonical sense. So our input for this model would be a genetic sequence centered around a potential start codon. Here we have an ATG. Pass it through layers of a deep learning model and get a final prediction. This prediction would be either the strength of translation initiation for that start codon or the probability that that start codon is a site of translation initiation. And when I talk about a probability, we get a number between 0 and 1, where a prediction above 0.5 means that our model is classifying that as an initiation site, whereas a probability below 0.5 means our model believes it is a skipped codon. It's not a site of translation initiation. So what does that look like? Here we have this SDHB gene. Here we see the annotated or canonical initiation site. It's an AUG codon. And in our sequencing data, we see that there is lactamyomycin enrichment. So if we give our model these 4 potential start codons, we want a classification of 1 at this codon and 0s at the other 3. When we train a model in this fashion, we're able to kind of recapitulate what is seen in literature before and quantify how translation occurs in optimal genetic context. So over on this figure on the left, we have our model prediction in log odds now. So if you take a probability of 0.5, you convert it to log odds, that will be 0 in log odds. So anything above 0 log odds means our model's predicting that that is a translation initiation site. When we do this, we see that a lot of AUG codons are predicted as initiation sites, whereas strong close cognates, which would be CUGs, GUGs, ACGs, the close codons that have a lot of literature out there to support their initiation, are much weaker, but still have some initiation sites. And then the other AUG variants are very weak, with only a few being initiation sites. Now, if we look at the annotated translation initiation sites, we see that these are very strong compared to AUG UORFs and other non-canonical translational events. This should be expected, and it's good that we can kind of delineate this because throughout evolution, your annotated translation initiation sites should be your most optimized sites, your strongest sites. This produces the proteins that are, essential for all functions in your cells. And if we look at a histogram of plotting these annotated initiation sites, we can see that, they get very strong predictions. And then we sample a random number or the same number of random genomic locations throughout the genome. We see that our model generally predicts all of these as 0, which again should be expected. These should be really weak predictions. Now, we can see kind of the behavior of our model, but we want to know why is our model working like this? What sort of other genetic features do we not know about that could exist that drive model predictions? To do this, we'll take a strong initiation site, take this AUG here, and we'll look at the genetic sequence that sort of flanks it upstream and downstream. We will manipulate or disrupt the motifs that exist upstream and downstream by replacing them with random nucleotides. Since this is a strong initiation site that we know of, there will be some genetic features that should be causing that to be a strong initiation site. And when we disrupt those, our model prediction should decrease drastically, giving us an impact of these features. When we do that, we see that there's GC-rich motifs that appear to impact translation initiation on a large level far away from start codons. In non-canonical translation, U-rich motifs seem to impact translation. So we need to do more work to figure out what this actually means. But nevertheless, these are features that seem to impact translation in a range farther away from start codons than most people think. Now we want to develop an expanded deep learning model to learn about translation across full genes or transcripts. So what would this look like? Okay, so at this SDHB gene in the first model, this ATG was the center of a sequence that was the input for our model. Now, what if the input was this entire genetic sequence of the SDHB gene and our model predicted which nucleotides were initiation sites along this gene? What this would allow us to do is we not only learn about start codon composition the sequence context. We also learn about how the 5' position of start codons affect translation initiation, and how the presence of upstream open reading frames affect translation initiation. So the necessary updates to make this model possible would be our input has to be 1,500 nucleotides long. Why do we pick 1,500? This is the kind of middle ground between computational efficiency and practicality. 19,000 of your 20,000 protein coding genes have annotated initiation sites within these first 1,500 nucleotides. And then the output has to be 1,500 nucleotides long to have where each output, each individual like index is the probability that the corresponding nucleotide is an initiation site. So as you can imagine, if we look at this gene, many of these locations are 0. They're not initiation sites with only one initiation site. Therefore, this data is very sparse. So to handle the sort of sparse nature, we'll use a transformer recurrent unit, which uses the attention layer to handle sparse information. And then we'll look at we'll beef up our input by using AIM-1 model predictions, the one I just talked about, and the density of annotated initiation sites to add bias or prior probabilities to our model to help it learn. After doing that, we can see that our model are sensitive to all the features I just talked about. And here we see that it's sensitive to codon positioning. So in the blue boxes, as the model, as the AUG codons kind of go down the model 5' to 3', we see that their predictions decrease. Whereas our first model is more agnostic to codon position. While it learns this sort of dependency, it still learns the importance of the genetic sequence context. Annotated translation initiation sites are often not the first AUG in the transcript. However, these are predicted with the highest confidence in our new model. And then if we look at the genetic sequence around the strongest predicted AUGs and compare that to the genetic sequence around the weakest predicted AUGs, we see that the strongest predicted sites in our model have the Kozak sequence present. So this model still learns all of these features. Now, how can we kind of apply this? Well, we can apply this by looking at clinically relevant variants and seeing if our model is sensitive or if translation is sensitive to these specific variants. So here, I again bring up this SDHB gene. There's a pathogenic variant in the ClinVar database that's associated with predisposition to hereditary cancers, where the ATG, the canonical annotated ATG, is switched to an ATA. Our model is sensitive to this and says that this is no longer an initiation site, but a skip site. This gene is a tumor suppressor gene, and when it is not being produced at the right levels, it no longer prevents the cells from growing in hypoxic environments, which is why we can say, okay, this could lead to cancers, right? That's a hallmark of cancer. So this should be very intuitive that this is happening. If you take out the start codon of a very important gene, you're obviously going to have problems with translation. But the important thing to note is our model is sensitive to these sort of variants, and we can get a quantitative answer for what these variants are doing. So now this is kind of work in progress, but we can take a subset of clinical variants and we can see which one of those variants seem to be affecting canonical or non-canonical translation throughout the entire transcriptome. And we see that some variants seem to drastically change our model's predictions in silico that are far away from start codons. So now we can look and say, okay, we have variants that are clinically connected, but some of these should now be affecting translation as well. So we can kind of see a pathway to disease. So in AIM-1, we looked at local genetic features that drive translation initiation. In AIM-2, we try to look at the context of translation initiation throughout full-length transcripts. And then in the future, I'm finishing up AIM-3, which is trying to look at these variants that affect canonical and non-canonical translation and also affect disease to try to understand disease progression better. So I want to thank all the people in both of my labs for helping me with ideas and carrying out this sort of project, give me advice, and thank you for the PGTP as giving great environment to learn and do work. So thank you for all of your attention, and please feel free to ask any questions.
All right, thanks, John. Appreciate it. Does everyone have any questions for John? Yes. How about you, John? Are you going to get a chance to head over to Gemma's lab and check out some of her work as well?
So yes, I think that's part of what we have to do. But it's like I think what I'd reach out and try to talk to her about is like the only way to kind of validate anything I show here is through like reporter assays of some kind. And that I don't know exactly what her lab like has in mind or uses for that. I have to do more work on that like myself. But that sort of wet lab experience would be kind of why her and I talked, and she was my co-mentor here. But that's a step if I really want to make this like really